welcome to the 10th TA session for Drug Delivery Principles and Engineering. And today also we'll discuss the topics that were covered last week and subsequently solve some previous year questions based on these topics. So these were the topics that were covered in week 10. Vaccines, which includes immuno-isolated cell therapy and gene therapy. So vaccines are basically used to induce an immune response or an immunological memory against a target pathogen or disease associated molecule. There can be two types of vaccines: prophylactic, that helps in prevention of a specific infection, or it could be therapeutic, that enhances the immune response against an existing disease. Now this vaccine could be of different types. It could be either live attenuated organisms, or it could be an inactivated or killed pathogens, or it could be a subunit of the pathogen or a recombinant polysaccharide or conjugate from the protein or a component of the pathogen. It could be toxoid, that is it inactivate the toxins rather than the organisms, or it could be a DNA or RNA vaccine which so that the host cells express antigenic proteins. These antigens and some adjuvants are then used to deliver these to the antigen presenting cells for a T and B cell activation, which then generate a immunological memory for immunological memory that will help you in future attack by the same target pathogen and help in enhanced immune response. So particles or microparticles could be used to deliver these antigens and adjuvants for optimized uptake by immune cells. And these particle based can be used for DNA or mRNA delivery or cancer vaccines, etc. Now this immune isolated cell therapy, so it's basically transplantation of cells that secrete therapeutic proteins while being physically isolated or encapsulated from the host's immune system. And the mechanism it follows is cells that are encapsulated in a semi-permeable polymeric membrane. It could be a hydrogel or a caged polymer, and the membrane then allows passage of nutrients, oxygen and cell products, but it will block immune cells and other large immunoglobulins from getting inside this cage membrane where the cells are residing, and thereby it prevents immune rejection. A major advantage of this is it enables transplantation of allogenic or xenogenic cells without lifelong immunosuppression. And these implant types could be either in macro or micro forms to encapsulate small number of cells or larger mass of cells, and it could be also vascular grafts to directly connect to host vasculature. Next is the gene therapy. It is treatment of diseases by introducing a genetic material, a nucleic acid, which could be either DNA or RNA, into a patient's cells. So it replaces missing or defective genes, or it could repair faulty genes, or enable cells to produce therapeutic proteins, or vaccinate against infectious agents by expressing immunogenic proteins. So there could be cell-based or direct gene delivery. The cell-based approach uses genetically engineered cells which are encapsulated and implanted in vivo, and these secrete therapeutic proteins. Whereas direct gene delivery involves nucleic acids like DNA or RNA, which is delivered directly to target tissue using viral or non-viral vectors. Viral vectors have like lentiviral or retroviral, have high transfection efficiency but risk immune reaction and insertional mutagenesis, whereas non-viral carriers have low immunogenicity and toxicity but are less efficient. So, based on these topics, we'll now see some previous year questions. Here is our first question. Which of the following approaches is not suitable to improve the biocompatibility of polymeric implants? And the options are coating with hydrophilic polymers, coating with hydrophobic polymers, peg coating, reducing surface roughness, or making the material more porous. Okay, so the correct answer here would be coating with hydrophobic polymers. So, hydrophobic polymer surfaces generally result in poor biocompatibility because they tend to support undesirable protein adsorption and activated monocyte adhesion, which can trigger further local inflammatory responses and fibrous tissue formation around the implant. So, coating with hydrophilic polymers or PEG will enhance protein resistance and reduce inflammation, thus improving biocompatibility. Moreover, modifying the surface to reduce roughness or making the material more porous also enhances integration and tissue compatibility. Therefore, option 2, that is coating with hydrophobic polymers, is the least suitable method or is not suitable to improve the biocompatibility of polymeric implants. Moving on to the next question. Which two of the following statements about vaccines are false? You have to select two correct options here. The options are: a therapeutic vaccine is given before manifestation of the symptoms of the disease. A prophylactic vaccine generates a memory for epitope of a pathogen in the immune system. Option 3, the polio vaccine is a prophylactic vaccine. Option 4, a prophylactic vaccine is given after the disease has occurred. Option 5, the purpose of a prophylactic vaccine is to strengthen the immune system to fight against the disease. Which of these two are false statements? So the correct answer here would be option 1 and uh, option 4. Option 1, that is a therapeutic vaccine is given before manifestation of the symptoms of the disease. And option 4, that a prophylactic vaccine is given after the disease has occurred. So a therapeutic vaccine is usually given after the disease has occurred, while prophylactic vaccines are given before the disease has occurred. So this prophylactic vaccines helps in generating a memory of for the epitope of a pathogen in the immune system and helps enhancing the immune response against in case of the actual disease. Whereas therapeutic vaccines are generally administered after the disease has developed, so to fight against that ongoing disease. Moving on to the next question, which consider that the following molecular species are contained in a dialysis bag with a 50 kDa cutoff. Which of the following molecules will be retained inside the bag? You have to select two correct options here. The options are dissolved oxygen, glucose, free radicals, nutrients, complement factors, IgG or IgM. Correct answer here would be IgG and IgM. These are usually of the molecular weight of 150 to 150 kDa and 900 to 950 kDa, which are way above the molecular cutoff of 50 kDa given in the question. So these only these two molecules will remain inside the bag. All of the other molecules may not be contained. Dissolved oxygen, glucose, free radicals, and nutrients are usually much smaller than this and can easily pass through the uh, dialysis bag. Complement factors are mixture of molecules, and while some of them could be higher than the cutoff, but if we look at the overall complement factors, the entire complement factors cannot be retained inside the dialysis bag. So only IgG and IgM will remain inside the bag, and that will be the two correct answers. Moving on to the next question. Tetanus vaccine is an example of a. Options are live attenuated vaccine, inactivated vaccine, conjugate vaccine, toxoid vaccine, recombinant vaccine, or polysaccharide vaccine. The correct answer here would be option 4, toxoid vaccine. So the tetanus vaccine is created using the inactivated tetanus toxin or a toxoid, not the live or the killed bacteria itself. And this toxin is chemically treated to remove toxicity but retain its ability to stimulate an immune response. These are generally used to design to protect against diseases caused by bacterial toxins by generating immunity to the toxin rather than the infecting organism itself. And tetanus vaccine is one such toxoid vaccine. Moving on to the next question, which of the following is characteristic features of immuno-isolated cell therapy? Options are: the immuno-isolated cells have a polymer encapsulation which allows nutrients and small molecules to reach cells but not antibody molecules and T cells. Option 2, cells are genetically altered to produce and secrete high level of toxins. Option 3, it involves replacing a missing or defective gene. Option 4, it repairs a malfunctional or non-functional gene. Correct answer here would be the immunoisolated cells have a polymer encapsulation which allows nutrients and small molecules to reach cells but not antibody molecules and T cells. This is the characteristic of immunoisolated cell therapy. Usually this antibody are very large molecules as compared to the small molecules and nutrients. So these are encapsulated in polymer with smaller pore sizes to let only the nutrients and small molecules to flow out, but then not the antibody molecules to pass through those pores. Moving on to the next question. Which of the following is a type of therapeutic vaccine? Options are MMR vaccine, cancer vaccine, polio vaccine, HCV vaccine, or smallpox vaccine. The correct answer here would be option 2, cancer vaccine. So, a therapeutic vaccine is usually administered to treat an existing disease, not to prevent its occurrence. And cancer vaccine are a classic example of these therapeutic vaccines because they stimulate the immune system to target cancer cells already present in the body. In contrast, MMR, polio, smallpox, and most HCV vaccines are prophylactic, wherein they are given to healthy individuals to prevent future disease and not for therapy after disease diagnosis. Okay, moving on to the next question. Which of the following are wrong about particle based vaccine methods? You have to select two correct options here. They are generally unstable and cannot be stored for a long time as compared to native antigen. Option 2. They are generally stable and can be stored for a long time as compared to native antigen. Option 3. Uptake of proteins can be higher when delivered using a nanoparticle. Option 4. There are extreme immune responses generated against the particle used in this method. Option 5. The antigen might lose integrity in the process of particle encapsulation. The correct answer here would be option 1. They are generally unstable and cannot be stored for a long time as compared to native antigen. And option 4, there are extreme immune responses generated against the particle used in this method. These two options are wrong about particle based vaccine methods. Particle based vaccines are designed specifically to improve stability of the protein or antigen cargo, and often making such vaccines much more stable and suitable for long term storage as compared to the free or native antigens. And while these nanoparticles and virus like particles can act as adjuvants and enhance immune responses, they are typically optimized to induce robust but not extreme or harmful immune responses. And most data show that these carriers safely modulate and enhance immunity without causing excessive or dangerous immune activation. So, option 1 and 4 are the correct options here, and they are wrong about the particle based vaccine methods. Moving on to the next question: what is an adjuvant in the context of immune response? The correct answer here would be option 5, a molecule that stimulates an immune response to target antigen. An adjuvant is specifically defined as a substance that enhances, increases, or modulates the immune response to an antigen in a vaccine. And adjuvants help in the immune system launch a stronger and longer lasting response to the target antigen, ensuring better vaccine effectiveness. So, option 5 would be the correct answer. Moving on to the next question: which of the following are undesirable properties of a polymer used for an immuno-isolated implant? You have to select two correct options here. Options are non-erodible, mechanically stable, toxic, biocompatible, biodegradable. The correct answer here would be option 3 toxic and option 5 biodegradable. So these are the two undesirable properties of a polymer used for an immunoisolated implant. Toxicity, because toxic polymers can release basically toxic compounds or harmful substances that can cause cell death, inflammation, or further systemic reactions and can lead to implant failure. So the polymers used for this immunoisolated implant must be non-toxic to both the encapsulated cells as well as the host cells and tissue. And next is the biodegradability. So the main goal of this polymers used for the immunoisolated implant is for the implant to be stable enough and non-degradable for long term. So the cells caged in this stable polymer will be retained inside and will continue to release proteins and exchange nutrients and molecules, rather than if they get degraded, then this encapsulated cells will get exposed to the host tissue and can elicit unwanted immune reactions. So the correct options are toxic and biodegradable. These are undesirable properties rather than you want non-toxic and non-biodegradable polymers that could be used for immunoisolated implants. The next question is, which of the following is a potential disadvantage of using particle based approaches while delivering vaccine to antigen presenting cells? There are two correct options here. The options are: the formulation can be unstable, it allows local delivery of antigens, the formulation can increase the in vivo half life of the vaccine, all kinds of antigens, proteins, peptides, DNA can be loaded. Option 5, co-delivery of adjuvants is possible. Option 6, APCs can take up particles in large quantities and might cause the cells to burst. Option 7, the antigen might lose integrity in the process of encapsulation. correct answer here would be option 1. The formulation can be unstable, and option 7, the antigen might lose integrity in the process of encapsulation. So this particle based approaches, these are the major concerns, that is formulation instability, because it can lead to unpredictable release, antigen loss, reduced vaccine efficacy, or difficulty in storage. And antigen integrity or loss can occur due to harsh chemical or physical processes used during the particle formation to encapsulate these antigens, and it could lead to structural changes, denaturation, or reduced recognition by immune cells. This compromises the ability of the antigen to elicit a proper immune response. So, the instability of the formulation or risk of antigen integrity loss are most recognized disadvantages of using particle based approaches while delivering vaccine to antigen presenting cells. So, that was all the questions I had for today, and see you all next week for the next TA session.